Right. Hello. Um, Today we are going to talk about the application of 16S RNA sequencing in wildlife conservation. I not only hope that you find this study as interesting and and honestly as critical as I did, but I also hope that you walk away knowing a little bit more about the Tasmanian devil and conservation efforts that we humans have used or are using, I guess I should say. The title of this talk was intended to foreshadow the bottom line of the study, but I'd also like to clearly state it here. The authors of this paper sought out to determine if monitoring of the gut microbiome would be a beneficial tool for conservationists involved in species restoration programs. So you may have noticed this first image is actually from a study conducted in 2015 by Cheng et al. Over the next few slides, I will discuss this earlier paper in a little bit more detail, just to help lay the foundation for why the authors of my chosen paper decided it was important to pursue more data in regards to changes in the gut microbiome of Tasmanian devils post-release into the wild. And you know what I enjoyed about this initial paper is that that they sought to define if the devil microbiomes differed when comparing wild populations to captive populations. And as you can see, they looked at a wide variety of different microbiomes. Um, and so I highly recommend giving this paper a read if you have the time. But from here on out, I will certainly be focusing on the gut microbiome. So what they did find in this first paper was that the ratio of firmicutes to bacteroidetes significantly differed when looking at captive animals versus wild animals. So it's interesting to note and to learn that wild populations have a higher ratio of firmicutes to bacteroidetes. This is a ratio that's also known as the obese microbiome. So typically you see the alternate, but in particular for the Tasmanian devil, this high ratio of firmicutes, which you can see represented there by the large chunk of blue, versus a small ratio of bacteroidetes, which is pretty much non-existent here on the chart, is really important for their diet. So this particular ratio correlates with efficient energy intake and storage from the diet. So in other populations, it can lead to obesity, but for the Tasmanian devil in particular, it supports their eating habits where they gorge nearly half their body weight and then they don't eat for many days. So interesting to see, that ratio kind of charted out here. And also important to note that this particular table and graph is showing that, a compilation of captive and wild together. So in the wild, sometimes you'll actually see 0 bacteroidetes, but here you do see that low percentage. So it is valuable, most definitely, to learn a little bit more about the devil. So they are carnivorous, nocturnal marsupials that were restricted to the island of Tasmania. I find that kind of interesting as I learn more about marsupials, thinking about the possum being the only native marsupial to the United States that I am aware of. And here we are seeing another one that's sort of restricted to one location. Anyways, despite being solitary, they often will flock to the same feeding grounds at the same time. So this sort of unusual behavior is really critical given this next development, which unfortunately was, in 1996, this devastating transmissible cancer known as devil facial tumor disease, or DFTD, was discovered in the species. It's one of a small number of contagious cancers ever recorded. So it's really unfortunate that it hit this population. This cancer is transmitted via biting and results in rapid growing facial tumors. You can see there in that image that I took off of the Save the Tasmanian Devil program website. The, um, the tumors themselves are, again, rapid growing and ultimately lead to starvation and the deterioration of normal bodily function, and fatality is pretty quick. So um, the interestingly, the primary lineage of this somatic cell disease is known as DFT1, and it was found to have female origins. However, in recent years, a new lineage, DFT2, appears to be a clone with male origins. So, in short, because this disease is so rapidly fatal, it reduced the population of sexually mature animals significantly, essentially decimating the population by nearly 80%. In 2008, the devil was declared endangered. And to this day, and as you can see as it's evolved a little bit here, the disease is very prevalent still within the wild population. Beginning in 2003, the Save the Tasmanian Devil program has been tirelessly trying to conserve the species by managing what they called insurance populations. So, in short, these populations represent different genetic lines, and they're housed in captive, intensive captive locations, as well as free-range enclosures. For perspective, intensive captive enclosures are much like a zoo, whereas the free-range enclosures are much larger and more closely mimic wildlife on Tasmania. It's important to note that these intensive captive environments allow for a highly regulated diet as well as opportunities for selective breeding. So also, make note, neither of these locations are infected with DFTD. So, these controlled populations are really here to supplement the wild populations that are still inflicted with the disease, and they're used as repopulation. And then at the same time, this repopulation it also prevents inbreeding, which is of course beneficial to the Tasmanian devil. So just briefly, in closing of this 2015 study, here's another figure, and it's illustrating the significant difference between the intensive captive gut microbiome compared to the free-range and wild incumbents throughout the island. In this image, you can see several wild populations represented by collections occurring at different locations throughout the island, donated by the GHNANTA. Using the Chow-1 verification measure, which is a statistical means of adjusting for differences in sample sizes by determining the smallest sample group and omitting reads from the larger groups. Researchers are able to show a statistically significant difference between the intensive captive cohort and the wild cohorts. Further, it shows a closer similarity between the free range and the wild, as well as zero significant differences between the multiple wild populations. And then to the right of this image illustrates the beta diversity of the gut microbiome across these cohorts. So they utilized unweighted unifrac, which highlights overall presence and absence of abundant organisms. And it illustrates sample groups that have less abundant organisms. So from this image, you can see clustering of the intensive captive population in the lower right-hand corner. This is illustrating that this population in particular has low diversity and also differs significantly from the other groups. Why today's paper? And I think this previous study was just a precursor where they identified a significant difference, but now they're begging the question whether or not the gut microbiome of Tasmanian devils are dynamic. So the questions tied tightly to disruptions associated with conservation efforts, such as, does the microbiome change over time after the host is translocated? And if so, how does it do it? And then do released individuals reacquire, a wild type microbiome after translocation? So to determine answers to these questions, the researchers studied two different translocation events. One included a captive to wild cohort, while the other included a wild to wild cohort. And I would like to draw attention to the small cohort sizes. So you can see, the captive to wild included 8 species, and then the wild to wild included mere 17. Um, so I guess I'm just curious if these groups should have been larger. Kind of hemmed and hawed at that question. Um, and curious thoughts on that matter. And then I've also included here just briefly how the fecal samples were collected and when. Please also take note of a difference between these studies whereby the wild-to-wild population included one seemingly extra sampling point, which took place after their brief stay in a free-range enclosure prior to release into the wild. Um, I'm assuming that the researchers were curious about the shift in the gut microbiome from a wild enclosure to a free-range enclosure back to a wild enclosure. But I guess, how do you all feel about them not considering that even an intensive captive cohort may have displayed differences that they did not capture by not sampling them when they were released from the free-range enclosure? And I will point out here in just a little bit, but both of these cohorts were held for 6 to 12 weeks within this free range enclosure, as is mentioned there at the top of this slide. I thought it was kind of neat to look at the supplemental data provided for this study. And this slide, as well as the next slide, show different housing or housing differences. So this first is the captive to wild translocation event. And I just highlighted differences that I thought were worth noting, but of course there's a lot more detail here. So mainly notes are in regards to the confinement or closeness of the animals. And then I also made note that Maria Island is considered a wild enclosure, but it is not Tasmania, so it is not their native island. And then further, here's the wild to wild translocation. And I didn't make note of too many differences here, but I did think it was interesting that they used two different locations for their free range enclosure periods. And I'm not sure if that is significant or not, but saw that, wanted to make note of it. So this first figure in the paper simply illustrates the sampling events in comparison to their overall timeline. I did find it interesting, in addition to that sampling point at the end of the free-range enclosure stay, there's also a longer timeline for the wild-to-wild in comparison to the captive wild. And then, of course, you can see it, they were sampling at different points within both of these studies. But I did like this visual. It gives a solid understanding of how this study took place. In regards to the methodology, researchers extracted DNA from the core of each fecal sample and generated barcoded amplicons of the 16S RNA. They used Illumina sequencing and the QIIME2 with the Data2 plugin to identify exact amplicon sequence variances, or ASVs, present in each sample, and then they filtered out the low abundance reads. Similarly to the previous study, they rarefied their data for diversity analysis with a minimum sequence being 3,838 for the captive to wild and 5,530 for the wild to wild. The taxonomy of the ASVs are assigned by comparison to the Silva 16S database. And then when we look at the data in the next few slides, I'll talk a little bit more about the tools they use for the alpha and beta diversity that are listed here. Um, in this paper, I will start by saying they documented these two study groups via two different figures. So they have figure 2 highlighting all of the charts for the captive to wild, and now they have also figure 3 for the wild to wild. As I started digging into this paper, I found it a lot easier to lay them out side by side. So for the remainder of this presentation, you will notice that I did that for you all as well. So I thought it was also interesting to note that in regards to the captive to wild cohorts, they compared their 8 captive devils to 7 wild incumbents. And so also the two dark red circles demonstrate, or at least to me demonstrated, that they did in fact choose wild incumbents in the second study compared to animals that have been released to Maria Island, because you can see some similarity there in regards to the baseline of those two cohorts. So we've got the wild population and the captive to wild there on the far right, and then they chose from that island another group of animals to then transport to Tasmania in the end there. So I thought that was kind of nice to see and make note of. Across all the samples collected in this first cohort, firmicutes dominated, followed by fusobacteria, proteobacteria, bacteroidetes, actinobacteria, and tenericutes. I did find it interesting that we see a spike in the bacteroidetes within the 6-month post-release captive to wild, as well as the free-range enclosure to wild. I'm kind of curious if this correlation is demonstrating something occurring, early on in a translocation process. Not sure. And then in regards to the wild to wild gut phylum, some similar phyla were represented with a return to higher diversity noted in that post-release 3 sample collection taking place 55 to 56 weeks after translocation. So you'll notice there's still differences, but it's getting back to the wild populations. So looking specifically at the alpha diversity, or the diversity within each cohort, the researchers used Chow-1 to determine statistical significance. Much like the earlier study, this method assessed richness and evenness by accounting for the sample size. So as expected, the wild incumbents showed the highest level of diversity, while the intensive captive demonstrated the lowest. Interestingly, in the wild to wild cohort, overall there was not a significant difference between the initial wild to the final wild, but there was certainly a statistical and significance between period of free range enclosure housings to the post-release group. And then again, I just made a note, these animals were housed in these free range enclosures for 6 to 12 weeks. And then after release, the samples were collected, 3 to 6 weeks, respectively, a little bit further, perhaps. But thus, I think it's showing this change in alpha diversity is occurring in a rather brief period of time. And I thought that was pretty interesting. And then to determine, if the gut microbiome changed significantly after release, the researchers looked at the beta diversity across the cohorts and utilized a Permanova test to determine statistical significance. I was not familiar with the Permanova calculation prior to this paper, so I can tell you that it looks at the diversity within a group and compares it to the diversity across groups, thus concluding if the variations seen are significant across the groups. So then they also used unweighted uniFrac graphs to show significant separation of the pre-release group from the post-release, and and the wild incumbents. So you can see that in the lower right-hand corner there, circled in red. And then within the wild to wild study, the post-release group showed significant separations from the pre-release and the free-range enclosure groups, shown by that obvious clustering in the lower left-hand corner. Um, And then one final look here, figure 3D, with the wild to wild, like I had mentioned earlier, they took another sampling at approximately 1 year post-release. Interestingly, these researchers looked at 3 additional fecal samples, and then what they see is that those samples did not show statistically, significant changes in the alpha diversity, but they did reveal close beta relationships between that post-release 3 group and the wild incumbents. So you're really seeing this return or change towards gut microbiomes that are similar to the wild type microbiomes in that area. So, in conclusion, these researchers found answers to their initial questions. Yes, the gut microbiome can shift, and when it does, it likes to return to levels similar to the local populations. Further, I'd like to point out that the shift occurred rather quickly, like I mentioned. I was surprised by that. I often think back to conversations about the first 3 years of human life, outlining the foundation of our gut microbiomes. So this shift was surprising. The researchers included a list of likely contributors to this change. And lastly, it was interesting to note that the wild-to-wild translocation did not result in increased microbiome diversity. I appreciated the logic that they used to explain this, keeping in mind that Maria Island, it's not native, but it is considered wild. So this particular location does have, different diet or, available species for these animals to feed on. So I think, as they mentioned, we're really seeing that they went from a rather lucrative area back to their native that doesn't have the same level of diet diversity. And then, as with most research, we find one answer and it leads us down the road of more questions. So, one point of review for this research in particular is, of course, the sample size that I've alluded to throughout this presentation. I don't know if there are general thoughts on this. And also, has anyone considered the role of the statistical tools used when drawing comparisons and conclusions? I know I learned a lot about them, but are they reliable? Do they introduce a bias? How do we decide which ones to use? I really thought that was an interesting point of this study. And then, as was briefly mentioned in previous slides, the researchers highlighted the role of both the environment as well as the diet. Was observed shifts in the gut microbiome. So, do both of these variables influence the gut microbiome equally, or does one weigh more heavily on the other? And then 2 final points. I enjoyed their further interest in studying the role of birth origins and multi-generational studies. I say this because, unfortunately, DFTD is not subsiding within the populations. So I do hope more research into the disease with possible eradication options arise. But in the meantime, it appears these insurance populations really are preventing the devil from going extinct. So as this program goes on and on, what changes are we going to see as more and more animals are birthed in captivity? And then lastly, I also appreciated their interest in comparing healthy and unhealthy devils. This may bring to light treatment opportunities. However, eradicating cancers is proven to be, pretty difficult. So here's just a list of cited literature. I'd also like to make note that there's wonderful supplemental tables within the paper. So feel free to dig around the raw data there. And then additionally, here is just a link to the Save the Tasmanian Devil program website. So thank you for listening. I hope this was enjoyable, and I look forward to discussing it with you further as the conversations continue. Thank you.